Thank
Okay,
you.
excellent. Thank you. Sorry if there's a little bit of background noise here, but I'm really honored to be invited to speak today and to get into it. I really just want to touch on the core focus of my lab to really understand the concepts of cell identity. And this is a piece that I wrote several years ago now on the evolving concept of cell identity now that we have these single cell technologies. And it was a really difficult piece to write because none of the reviewers could really agree on defining cell identity. But we attempt to do this in the lab through developing new technologies, and we work on several systems. So from development across several species through to differentiation across several systems, and I'll touch on adipogenesis today. And we focus heavily on reprogramming, and I'll discuss that in the first part of my talk. And we also apply these principles to disease, so specifically to pediatric intestinal failure. So trying to understand how we reverse cell identity changes in a disease context. So really here, the goal is the precise control of cell identity. So I'm breaking my talk into two parts today. So in the first part, I will discuss the new genomic technologies we've been developing to be able to deconstruct and inform the cell reprogramming process. And then in the second part of the talk, I'll switch to a new story for us. So really applying these methods to understand physiological, biological processes. And as I mentioned, I'll discuss our new work on adipogenesis. And so just as a broad overview on the cell engineering portion of this talk, we know that cellular reprogramming fails to recapitulate target cell identity. So we can start from differentiated cell types, reprogram them to pluripotency, and then take them out to desired cell types. This is quite inefficient. So as a shortcut on this map, we can take fully differentiated cells and try and directly convert them to alternate identities. But we know this isn't quite direct and that cells go back through these regenerative states, these developmental states. The problem in this field, though, was that there was no systematic way to measure cell identity, so the engineered cells to their in vivo counterparts. So 10 years ago now, we developed CellNet. So a bulk method to reconstruct gene regulatory networks and use those networks to be able to assess the cell types we were generating. And we found that directed differentiation, so taking cells to pluripotency and then out toward target cell identity, it was failing to produce cells that were developmentally mature. And this was really a bottleneck in this approach. It still is. Direct reprogramming, on the other hand, so trying to transit between fully differentiated states, we could see that the starting cell identity persists and the resulting cells are still developmentally immature, even though we're trying to avoid these developmental states. So now fast forward to 2022, so just a couple of years ago, and we released the Capybara tool. So this is a method to measure cell identity at single cell resolution. I'll go into more detail of this approach later. We found the same problems across the board, now just at higher resolution. The main point of this is to say that the practical utility of engineered cells is limited, and we want to know how we can deconstruct cell identity to be able to increase reprogramming efficiency and fidelity. Our favorite case study for cellular reprogramming is the conversion of mouse embryonic fibroblasts to induced hepatocytes, and this is via the overexpression of two transcription factors, FOXA1 and HNF4 alpha. So you get these epithelialized colonies emerging within about a week. We knew that these cells weren't quite induced hepatocytes, though. There were different signatures, underlying their identity. So we generated these cells, performed bulk expression profiling, ran that data through the CellNet platform, and it informed us that these cells actually have intestinal identity in addition to their hepatic potential, and they were able to actually functionally engraft the intestine long-term, in addition to their liver engraftment potential. So we renamed these cells induced endoderm progenitors, and it's a really fascinating cell type to study. But one of the challenges is only 1 to 2% of these cells would successfully convert to these induced endoderm progenitors, and then an even smaller number makes it through to fully differentiate to the intestine. So this is how we found ourselves in the field of single cell biology. So back in 2018, we profiled this reprogramming protocol So periodically sampling the cells every few days over a 4-week period. And doing this, we could break the reprogramming process into several distinct stages. So moving from fibroblasts through to these transition stages and out to a reprogrammed state. But what we couldn't answer with this approach was how we can pinpoint the origins of successfully reprogrammed cells. So for example, which of these fibroblasts will successfully make it through to this reprogrammed state, and what paths did they take through this transcriptional landscape? To address this and to generate ground truth lineage tracing data, because it's really a lineage problem here, we need to understand the origins of these successfully reprogramming cells. So to be able to trace lineage, we developed the cell tagging method. Now, this is a very simple lentivirus-based approach where we introduce random barcodes into cells. And these barcodes are followed by a polyadenylation signal. So they're produced as transcripts, and then they're captured as part of the single cell transcriptome. And then through periodic tagging and sampling of these cells and replacing, we can start to build rudimentary lineage trees. And the way we connect lineage here and connect clonal relationships is that cells, as they expand, inherit these barcodes, and based on the sharing of the same barcodes, we can define these lineage relationships. And from this work back in 2018, we found 2 conversion trajectories. So one to a reprogrammed identity and one into a dead-end state. So the reprogrammed induced endoderm progenesis from the fibroblasts. And so to get into more detail of these 2 states that we defined with the ground truth lineage, first of all, the reprogrammed state in terms of gene expression, we see upregulation of E-cadherin. So these are epithelialized cells. The dead end, on the other hand, these cells remain stuck in a mesenchymal state. So these are 2 quite distinct outcomes transcriptionally, but then functionally, how do these states differ? Because this is a very important point to address. So through flow cytometry, we were able to enrich for these reprogrammed and dead-end populations, and then we tagged them with defined cell tags and introduced them back into the mouse intestine that was acutely damaged in a competitive transplant assay. And then we found that it was the reprogrammed population that was statistically more likely to functionally engraft the intestine, and that these cells transitioned through a stem cell-like state. So here there's key differences between these reprogrammed and dead-end cells. So there's a desirability to push cells from this dead-end trajectory onto reprogrammed. So what more can we learn from this process? Like, how do we gain mechanistic, actionable insights to be able to switch their trajectories? So here we are looking at the clones that follow a successful reprogramming trajectory versus clones that move into this dead-end state. And what we can do is start looking at the paths they take through this landscape. And we found that the successfully reprogramming cells transition through this transient state where they upregulate the putative RNA methyltransferase mesyl7A1. And when we add mesyl7A1 to our FOXA1 HNF4 alpha reprogramming cocktail, we find that we produce more colonies by colony formation assay, but when we single cell profile these cells, we find that we're pushing more cells from this pre-reprogrammed state onto a reprogrammed trajectory, so guiding them to the desired outcome. So this is just a small vignette of how this ground truth lineage information can help us and provide insights into improving the reprogramming process. And so I think one of the most insightful results from the lineage tracing came from us being able to ask when reprogramming outcome is determined. So from this reprogramming lineage here, we have a clone of cells that were tagged as fibroblasts, so they've expanded. And then these sublineages in blue here derive from this common ancestor. They've been tagged again at day 3, so just after we finished overexpression of the reprogramming factors. And then out in yellow here is another round of cell tagging at day 13, so once the reprogrammed cells have emerged. And the dogma in the field has been that in the early stages of reprogramming, if you have siblings that receive the same amount of factors under the same conditions, that these early stages are stochastic, so that one subclone derived from this common ancestor may successfully reprogram, but another subclone may die. It may go on to turn into a completely different cell type. But because we've been able to define these reprogram and dead-end trajectories, we can actually take a look at those reprogramming patterns. And we find that clonally related cells share reprogramming outcome from the initiation of transcription factor overexpression. And now when we split siblings across independent biological replicates and wait for a few days to reprogram, we find that they no longer share reprogramming outcome. So this was a big result for us because it suggests that these trajectories are established at reprogramming initiation, so much earlier stages than we previously appreciated. And it also suggests that cells are transitioning through permissive states where they become malleable to reprogram. And mechanistically, that's really important because if we can push cells into these malleable states, we can massively increase reprogramming efficiency. To recap on this first part of the talk, the main takeaway from this is that reprogramming fate is determined at initiation and defined by these two trajectories. So one to the epithelialized reprogrammed state, driven by this transient upregulation of mesyl 7A1, and one route through to this dead-end mesenchymal state that we know doesn't efficiently functionally engraft the intestine. So we want to push more cells down this route. Now, the challenges of studying these initiation stages or the first few days of reprogramming is, first of all, in our original study, we didn't capture enough clones in these early stages of reprogramming. So these analyses were relatively underpowered. And the second issue is that transcriptome isn't everything. So we could see some transcriptional changes early on, but we know there's hidden heritable variables encoded in the epigenome that we need to capture to be able to understand this process fully. So this is where we introduce our next version of cell tagging, cell tag multi. So this project was led by a graduate student who's in my lab, Kunal. And similar to the original cell tagging, we can capture the cell tag barcodes, so the lineage in parallel with gene expression. But we've also rendered this approach compatible with barcode capture with chromatin accessibility, so single cell attack sequencing. So in parallel, we can capture chromatin accessibility, lineage, and gene expression at single cell resolution. And just to show you an example here of the efficient capture, here we're identifying different clones, and these are the barcodes captured across the independent assays. Very efficient barcode capture in this. And then from this co-embedding, we're just projecting a couple of the clones on here that we've been able to identify. So it's a very robust lineage tracing technology that works with epigenomic profiling. So we next applied this method to be able to link early gene regulatory state to reprogramming fate. And the question here was, what are the early gene regulatory changes that set cells onto a specific trajectory? So in this case, we take our mouse embryonic fibroblasts, we initiate our reprogramming with our two transcription factors. At the end of the process, we tag these with cell tag multi. We replate them, so we call this day 0. And then we allow them to expand for 3 days. So it's really important to have this clonal expansion to be able to capture these clonal relationships. And at day 3, we capture 50% of our cells to sufficiently power these experiments. And we term this our state sample. We then replate those cells, so the siblings go on to divide and reprogram, and we capture 2 further samples at days 12 and 21, and we refer to these as our fate samples. And what this allows us to do is capture these clones at day 3, so very early in the process, and we capture them and we can identify those clones that will eventually reprogram. We can also identify those clones that are destined to go into the dead end, and then reconstruct the early gene regulatory changes associated with these outcomes. And so in terms of looking at gene expression, and we can see that these day 3 reprogrammed and dead-end destined cells have differences in gene expression. But what personally I think it gets really interesting is when we start inferring transcription factor activity based on chromatin accessibility of the transcription factor motif. So we've used ChromeVar for this. And we see some stark contrast between the reprogrammed and dead-end destined cells at day 3. So on the reprogrammed destined cells, we see accessibility for motifs FOXA1 and HNF-RAVA, so suggesting, inferring that these transcription factors are active from early stages. And then we see a completely different pattern with our dead-end destined cells at day 3. And our most active inferred transcription factor here is ZFP281, so inferred to be active from early stages, specifically on the dead end. What does ZFP281 do? Well, we know from the literature that it drives a mesenchymal-like program in the context of breast cancer. And the key word there for us was mesenchymal, because we know the hallmark of the dead end is that they're stuck in this mesenchymal state. So to test this in further detail, we took our standard reprogramming protocol with FOX HNF4 alpha. Then we added ZFP281 in overexpression. We also knocked it down from the beginning of the reprogramming process and asked, how does this change the identity of the cells? And we find when we knock down ZFP281 that it shifts cells toward this successful reprogrammed identity, and it pulls them away from that dead end identity. So exactly how we'd anticipated. And this paper was published earlier this year, and we show that TGF-beta signaling is downstream of ZFP281. So this is what's biasing cells toward the dead end. And that when we block TGF-beta signaling with a small molecule inhibitor, we push even more cells onto this desired reprogrammed trajectory. So from this vignette, so combining single cell lineage tracing technologies with RNA-seq chromatin accessibility, what have we learned about the reprogramming process? So first of all, we know that the efficiency and fidelity is blunted by the generation of these off-target identities. And this is not specific to the IEP reprogramming process. We also observed this. Other groups have observed this in the context of reprogramming to pluripotency, cardiomyocyte reprogramming. So this seems to be a general feature. We know that we can redirect cells onto the successful reprogramming trajectory, so from these mechanistic insights. However, the efficiency is still low. And this is because the majority of reprogramming events originate from a handful of clones, and that cells are transitioning through these reprogramming permissive states. So this brings us to the hypothesis that in most cells, the target genes required to initiate reprogramming successfully are locked within heterochromatin. So really it's reprogramming outcome being determined by the gene regulatory state of the cell into which the reprogramming factors are introduced. So now we are shifting our focus to these very early stages of reprogramming. So watch out for a couple of preprints coming up in the next year on this subject. So shifting our focus even earlier in this process. So in the second part of my talk, as I mentioned, we've really focused on applying our methods to understand reprogramming. So you can think of this as quite a non-physiological process of forcing changes in identity via transcription factor overexpression. And we built all of our genomic tools, our analysis methods to be able to understand this process. But we also want to be able to build tools that we can apply to understand physiological biological processes. And this is what we've been doing in the last few years in the lab in the context of adipogenesis, so quite a new area for us. Adipogenesis right now is a really hot subject, so especially with the so-called miracle drug Ozempic, but that's really not the end of the story, because we know that with Ozempic, there are quite some undesirable side effects from this. We also know that patients tend not to stay on Ozempic long term. So we really are motivated to keep studying adipogenesis. Now, aside from obesity, fat is very much demonized, but fat is essential for healthy physiology. So white adipose really is central to regulation of energy storage, metabolism, immune response, hair growth, so has very positive benefits. So we set about trying to understand white adipose and adipogenesis. And this project has been led by two postdocs in my lab, Guillermo and Emily. And we pre-printed this story last year, but it's really been evolving since. And I'll present a lot of unpublished data on this today. So adipocyte progenitor commitment remains a black box. So we have our adipocyte progenitors that are marked by DPP4 expression, and then these transition into pre-adipocytes. So the cells cycle, they differentiate into this pre-adipocyte state marked by CD9 expression. From the pre-adipocyte state, they then differentiate to mature adipocytes. They leave the cell cycle, so cell cycle exit and full differentiation. So these later stages of adipogenesis have been well characterized, but these early stages of progenitor to pre-adipocyte transition have not been so well characterized. So we were interested in doing this. And we're interested in doing this in a specific depot. So what's fascinating about adipogenesis is that, there are lots of different depots across the body, and these are subject to differential regulation. So the depot we are specifically interested in here is the skin. So skin adipose has a naturally high production of mature adipocytes. So it's closely related with hair follicle cycle. And it's also closely related with the skin just being under environmental insult constantly. And now we compare the skin depot to the inguinal depot. So this is the fat around the thighs. So in comparison, this visceral depot is very sleepy. It's a much harder to shift depot of fat. So very, very different to the skin. So really trying to compare and contrast these two depots. So there's no existing single cell atlas on skin adipogenesis, and this is where we started. And so we took mice at P21 and telogen, and this was specifically to enrich for adipogenic progenitors, so looking at these early stages of the process. We also perform APC enrichment in here, and then single cell RNA sequencing. We want to avoid these mature adipocytes in this profiling because it's very difficult to perform single cell profiling on them. And the single nucleus profiling wasn't really compatible with our lineage tracing that I'll come to. But from this initial profiling, we collected almost 15,000 cells, which was sufficient to ask our initial questions of what is the heterogeneity of these progenitors, specifically in the skin adipose depot. So to start typing cells in an unbiased way, we used our capybara method. Now, capybara takes your single cell profiles and matches them to existing single cell reference atlases. So we made a mega reference of all adipose depots that had been subjected to single cell profiling. And we found that the closest matching reference for us was the inguinal adipose reference that was published by Merrick in 2019. So using this reference, we then assigned capybara identities of our skin adipogenesis atlas. And the first observation here is that most of the cells in the skin are progenitors compared to the inguinal depot. So really expanded progenitors pool in the skin. We also observed that we have this expanded population of these previously uncharacterized cells. So previously they were termed group 5. So really representing a tiny proportion in the inguinal tissue. So they were overlooked previously. They were thought to not be adipogenic and just some side population that remained uncharacterized. But with such an expansion in the skin and representing, a sizable population, we wanted to understand these cells in more detail. So looking at gene expression in these group 5 cells, it sat within this interesting hierarchy. So as I said, DPP4 expression marks the progenitors, and then CD9 expression marks the differentiating committed preadipocytes. And we found that these uncharacterized group 5 cells sat in between the progenitors and the committed preadipocytes based on this very, very simple scheme, just on two markers, but we get a lot more detail than this. So based on this gene expression, we propose that this group 5 represent immature preadipocytes, so a defined cell population that sits between progenitors and committed preadipocytes, so very early in this process. And now, thanks to the single cell profiling, we were able to identify cell surface markers that allowed for an enrichment strategy. And then using this enrichment strategy, so via flow cytometry, we were able to enrich for progenitors and also enrich for this immature preadipocyte compartment, and then transplant them back into mouse skin and ask, well, is this immature preadipocyte compartment actually adipogenic? Because previously it was thought not to be not to have anything to do with adipogenesis. And we do find that these cells can give rise to mature adipocytes based on perilipin staining. They also express markers of mature adipocytes following their transplant. So these immature preadipocytes that we're calling them are definitely part of this adipogenic hierarchy. But the question is this a linear process? And are the immature preadipocytes actually derived from this progenesis state? So this is where our cell tagging comes back in. So as I explained to you, lentiviral-based approach, we can tag cells and follow them, and in this case, in an in vivo model. So again, we take our cells, take our mice a P21, and we isolate by our flow cytometry scheme and the progenitors and the preadipocyte populations, and we tag them with defined cell tags. So this is similar to our competitive transplant assay that I presented in the intestine a few slides ago. We then introduce them back into the mouse skin in a competitive transplant assay, and then 11 days later, we recover the cells for single cell profiling. First result, so just focusing on this progenitor population. So we introduce the progenitors back into the skin, and we find they give rise to considerable populations of immature and committed preadipocytes. So we can confirm that these immature preadipocytes are derived from these progenitor cells and then go on to produce the committed preadipocytes. Now, where it gets particularly interesting is looking at this preadipocyte population. So you'd expect if you're putting in, so transplanting these more mature cells, that you should get even more differentiation from this process. And that's not what we find at all. We see that the pre-existing immature preadipocytes are less likely to differentiate. So they're possibly missing a cue in this process. So this was a very curious result to us. And it started to challenge the idea that this could be a linear process. So to present our refined hierarchy of skin adipogenesis first, our previous understanding was that progenitors differentiate to preadipocytes, and then they mature out toward mature adipocytes. And very little was known about these early stages. And our current understanding, based on our single cell profiling of this depot and our lineage tracing, we now understand that progenitors give rise to transitioning progenitors through to these immature preadipocytes, and then they go on to differentiate. So this is when we transplant the progenitors. So we think this is a linear process. However, when we transplant immature preadipocytes, they don't go on to fully differentiate, so suggesting they could be missing a cue. An alternate explanation to this is that it's not a linear process, so it could be a branching process, so that progenitors can directly give rise to committed preadipocytes, and they can also directly give rise to these immature preadipocytes. And that would explain the observation of why these cells are accumulating in this stage. So to test this hypothesis in more detail, so is this a linear or branching process, we can go to our cell tag data. So I said to you that we'd use these defined cell tag barcodes for the competitive transplant assays. These cell tag barcodes also contain the random portion that we'd used in our reprogramming experiments. So based on the sharing of these random barcodes, we can identify clonal relationships from this data set. Now, the first interesting observation is that relative to progenitors, there are fewer preadipocyte clones. So this is an important observation because it suggests that the preadipocytes after transplant, they are not dividing for some reason. And I mentioned this earlier, that the cells are dividing and cell cycle is linked to differentiation in this system. So cells have to be cycling to move forward into this preadipocyte commitment. So that was an interesting observation. The expanded preadipocyte commitment that we look at post-transplant, it's comprised of orphans. So these are not dividing cells, they're potentially quiescent and non-adipogenic. And so we can get further into this clonal analysis now, and it supports a linear model. So what I want to focus on here, so these are all the clones we've identified in our transplanted progenitors. So you can see a huge pool of clones. So all the clones remain in that progenitor state. But there's also a substantial proportion of cells here where clones are connecting by lineage the immature and the committed preadipocyte compartments here. So this rule, it does not rule out, but it supports a linear model where immature preadipocytes differentiate from progenitors, and then they can also give rise to committed adipocytes rather than the branching process. In the preadipocyte compartment, so this is a compartment where we can follow these clonal relationships because the cells have been dividing. When we see the cells dividing, we see them advancing through the differentiation process. And again, we see these lineage relationships between the immature and the committed compartments. So again, providing support to this linear model. But we do stress that we can't fully rule out this branching model. We also can't rule out that cells might regress into a progenitor state. So these lineages are extremely complex. One of the main points here is that cycling is required for this immature to committed transition. I'm going to come back to some of this mechanistically. So the next question is, what are the molecular drivers of adipogenesis in the skin? So to address this, we used our method called Cell Oracle. And Cell Oracle reconstructs gene regulatory networks from single-cell RNA-seq and ATAC-seq data. And then we use these networks to be able to perform in silico gene perturbation. So in this case, we would knock out in silico a transcription factor of interest, so set its expression to 0. We can also overexpress it, so within a physiological range, and then use these in silico models to predict how cell identity will shift following these transcription factor perturbations. So really, we're using these networks to be able to interrogate the regulation of cell identity. So when we perturb a transcription factor, based on expected changes in gene expression, how would identity be shifted? And using this method, so the main output of Cell Oracle are these simulation vectors. So they're predicting cell identity will move in this direction when we perform this perturbation. We look at how these perturbations are predicted to respond to impact differentiation in that natural differentiation process. So we look at how these differentiation and simulation vectors combine. So in this case, the differentiation vectors and simulation vectors are sharing a direction. So we'd expect that higher cell density in later stages of the differentiation process, so suggesting that differentiation is accelerated. In the case where the differentiation vectors and the simulation vectors are opposing each other, we'd expect cell accumulation earlier in the process, so suggesting that differentiation is inhibited. And we published Cell Oracle last year and performed a lot of validation in the context of zebrafish embryogenesis. So applying Cell Oracle to adipogenesis, we are specifically focusing on the progenitor to immature pre-adipocyte transition. So looking at these very early stages of commitment. So we first create this pseudotemporal ordering with our data, so using monocle 2, and then we calculate these pseudotime vectors. So this represents the natural differentiation of the system. And we then perform a systematic knockout simulation and overexpression simulation for all transcription factors within these networks, and then calculating the inner product between the pseudotime vectors and the knockout vectors. It produces a perturbation score. And the areas in pink here is where differentiation is predicted to be inhibited. The areas in green is where differentiation is predicted to be accelerated. And so using this, we can perform these systematic perturbations in silico, and Cell Oracle picked out SOX9 as a top predicted regulator of adipogenesis. Now, SOX9 expression is gradually upregulated as adipogenesis progresses, and Cell Oracle is predicting that differentiation will be arrested in this system. So compared to the control cell density, so the control cells are progressing nicely along this differentiation trajectory, SOX9 knockout blocks its differentiation. Interestingly, SOX9 expression downregulation is required at later stages of adipogenesis for cell cycle exit and full differentiation. So to test these predictions experimentally, we performed a competitive transplant in which we knocked down SOX9 and also overexpress it relative to control. Now, the challenge with this is that the SOX9 overexpressing cells completely outcompeted our control and knockdown cells, which was really interesting. So from doing some in vitro assays, we found that SOX9 knockdown inhibits proliferation. It also inhibits differentiation. So cell cycle and differentiation are intimately linked in these early stages. On the flip side, what happens if we simulate the effects of SOX9 upregulation in silico? Well, we find that Cell Oracle predicts that differentiation will be accelerated. So again, we perform these experiments to test these hypotheses. And we find that in SOX9 overexpressing cells, differentiation is accelerated as we predicted. But interestingly, SOX9 overexpression in the immature compartment does not advance differentiation. So this is really interesting. So again, suggesting that these cells have lost a cue, they've stopped cycling. They can't respond to SOX9 in this context, or there's already sufficient SOX9 in the system. So looking at our model of SOX9 here, we have this gradual upregulation into this immature pre-adipocyte state. Cells are cycling, they're differentiating through to the committed pre-adipocyte state. SOX9 is then rapidly downregulated, cells exit from the cell cycle, and they're able to differentiate to mature adipocytes. So what cues are these cells responding to in the skin? So we've been saying there could be a missing cue in these immature progenitors, but generally this is a very cell intrinsic view of cell identity. We want to understand more about the cell extrinsic regulation of this progenitor to pre-adipocyte transition. So to address this, we can perform cross-depot transplants. So here what we've done is take the progenitors from the skin and we've injected them into the inguinal depot, and we've also injected them into the skin depot and asked how they respond to these different environments. So the first observation we make is that we get an expansion of this committed pre-adipocyte compartment in the skin depot. This makes sense because rapid adipogenesis is required for the active turnover of the skin depot relative to the more dormant inguinal depot. And now when we look at the gene expression patterns, so to try and dissect what signals these cells are responding to, and we see that progenitors in the skin harbor this immune inflammation signature. So this is potentially what they're responding to this in the skin, what's driving their cell cycling and their differentiation in this process. So our current understanding of adipocyte progenitor commitment, so this previous simple linear model, has now got a lot more complex and really fun, we hope. So we see that the progenitors give rise to this differentiation process. But when we take these immature pre-existing pre-adipocytes, take them out of the system, put them back via transplant, there may be loss of cues, it stops them from cycling, stops their differentiation process. And we understand that, so from application of our methods, that SOX9 regulates the proliferation and differentiation of progenitors into committed pre-adipocytes, so these very early stages of the process. And we found through this cross-depot transplant that progenitors respond to specific cues in the skin. And with that, I would like to thank my lab, thank our funders. We recently moved to Harvard and Brigham, and we are looking for talented postdocs to join us. And with that, I would love to take questions. Thank you.